Once you have that secondary exposure is going to give you that very rapid, very efficient exposure that's going to initiate the whole response. And that whole response is going to come very quickly. It's going to be very amplified, and you're going to have this immune response taking place, and you may not even know that you have been infected. So, this is a response that will be very rapid and will give you protection right away. And this is going to be primarily due to the number of memory cells that we have. So your B memory and your T memory, they're going to go ahead. We don't have to have that whole antigen presentation anymore. We can initiate those B cells and those T cells, and you can, well, you do have to have antigen presentation, but you don't have to have that whole clonal selection, that whole maturation that takes that long period of time. We've got these memory cells now that can recognize that antigen and begin the whole process right away. Our cell-mediated immunity, we've just talked about our B cells and those antibodies. Now let's talk about our T cells. These T cells are going to require direct involvement of those T lymphocytes, and they are going to be the ones that do the job throughout the whole course of the reaction. You're going to have these T cells are going to come into contact with some MHC complex before they become activated. Once they get stimulated, you're going to have that one T cell is going to activate other T cells as well as the B cells and some phagocytes within the phagocytic system. They also are going to start to produce things like cytokines. Those cytokines are going to be chemical mediators that are going to turn things on, that are going to initiate the the cells to become activated, your T helper 1, your T helper 2, and your cytotoxic T cells. Once you have those T cells maturing, they've gone to the lymphoid organs, and they're going to be waiting there to react with their particular antigen. Once they recognize that antigen that's presented to them on that MHC carrier, those CD4 receptors are going to bind to those MHC2 receptors. The CD8 receptors are going to recognize the MHC1 receptors, and these T cells then are going to be bound to those MHC complexes, whether they're the CD4 MHC2 or the CD8 MHC1. The T cells are going to be activated, and they're going to be transformed through mitosis into our effector cells and into our memory cells. Again, go back to the picture on page 427 and kind of follow that through this whole process. Here's a subset of the T cells. The only ones that you are going to be responsible for are this T helper 1, the T helper 2, and the cytotoxic T cells. We're not going to talk about the regulatory or the T helper 17 cells, okay? These are just other cells. They don't have quite the same activation and the like, same function within the immune system. So T helper 1, T helper 2, and cytotoxic T cells. The T cells, the T helper cells, these are going to be those central cells. They're going to be the ones that are going to initiate that whole immune response. They're also going to help with the regulation of immune reactions. They're going to be involved in the receptor contact. They're going to activate those macrophages. They're going to have that those macrophages and those T cells are going to bind together. You're going to have the release of cytokines, and those cytokines are going to have a stimulation on other T cells to begin the whole complex. They're also going to secrete that interleukin 2. That interleukin 2 is going to activate the B cells. It's also going to have some activation on some of the other T cells. Don't worry about interleukins 4, 5, and 6. When you have these T helper cells that attach to those antigen MHC complex, you're going to have those T helper cells differentiate either into a T helper 1 or a T helper 2 cell. If you have a T helper 1, it's going to go one direction. A T helper 2 is going to go another direction, and they're going to have a coordinated effort in the immune response. The regulatory T cells, these basically just are those cells that are going to have some regulatory response. They do carry CD4 receptors. Their main function is to control inflammation. They also are going to help to prevent that autoimmune response, those self versus non-self irregularities. You can also have these regulatory cells that are going to help with the prevention of response against your normal flora. Your normal flora is not self-tissue, but it is bacteria that is good and carries on a very good relationship with you and your flora. So you don't want to have an immune response against your flora. Don't worry about too much about the regulatory cells. This is really kind of all you need to know. Cytotoxic T cells, these are going to be T cells that are not going to have an effect in the blood, but they're going to kill in specific target tissues. You're going to have these cytotoxic T cells, instead of having CD4 receptors, they're going to bind to they're going to have CD8 receptors. Because they have CD8 receptors, they're only going to bind to those MHC class 1 proteins. Those MHC class 1 proteins, remember, are on every cell in the body. MHC class 2 are only on the macrophages and the dendritic cells and the B cells. So if it's got a CD8 receptor, it's going to bind to MHC class 1. And when you have an antigen complexing with that class 1 MHC, these cytotoxic T cells are going to be involved in the immune response. When that cytotoxic T cell binds with the MHC class 1, you're going to have activation of that cytotoxic cell, and it's going to start secreting things like perforins and granzymes. These perforins and granzymes are going to be things that are going to basically punch holes in the membrane. Particularly perforins, these are going to punch holes in the target cell, and these granzymes are going to be enzymes that are going to be attack proteins that are basically going to cause the cell to be destroyed directly by the T cell itself. The T cell can destroy cells that are virally infected, that are in the tissues. They can destroy cancer cells, and they can destroy cells that are in the body from other animals and other humans. These cytotoxic T cells are the T cells that we have that are responsible for the transplant rejection. These are going to begin to start targeting those tissues that have been transplanted into a body that don't carry the same CD8 receptor, and so they're going to recognize them as foreign and start to attack them. And these are the cells that we're going to try with immunosuppressive drugs to kind of calm down. Here is the cytotoxic T cell activity. This animation does a really good job of showing you the difference between MHC1 and MHC2. Remember, MHC2 are going to be the macrophages and the dendritic cells. They're going to bind to CD4 receptors. These have CD8 receptors and MHC class 1.
When a virus or other infectious agent infects a cell, some of the proteins of the infectious agent are broken down into peptide fragments. These fragments are complexed with special proteins called class 1 MHCs, major histocompatibility complex proteins, and displayed on the surface of the infected cell. The T cell receptor on the cytotoxic T cells interacts with the virus-infected cells by recognizing both the foreign protein fragment and the class 1 MHC. The cytotoxic T cell then releases toxic substances which cause death of the infected cell. Cytotoxic T cells also produce a toxin called perforin, which creates holes in the cell membrane, causing the cell to break open.
So you see that the cytotoxic T cell actually works right directly on the cell, whereas the antibodies that are made from our B cells target the cell and then maybe initiate something like phagocytosis or complement. The T helper cells, they do the same thing. They're going to bind to that target cell and destroy that target cell or initiate the destruction of that target cell. Here is basically our T cell activation. If we look up here, this is going to be our T helper 1, T helper 2, and down here is going to be our cytotoxic T cell. So let's look at our antigen presenting cell here. This antigen presenting cell is our dendritic cell. It has an MHC class 2 protein on its surface. So that means any T cell with a CD4 receptor, particularly this T helper 2 cell, this T helper cell here is going to bind to this CD4 receptor, and it's going to go through cell division. It's going to make either memory cells, it's going to make T helper 2 cells, which are going to release interleukin. That interleukin is going to activate our B cells. Our B cells then are going to make either our plasma cells, our memory cells, or what we call B regulatory cells. For this lecture, please just focus on the plasma cells and the B memory cells. Once we've activated our B cells and they've gone through cell division, our plasma cells are going to then go and produce antibody. This T cell can also become a T helper 1. This is going to produce something called tumor necrosis factor, and this is basically going to stimulate macrophages to have some hypersensitivity. These will be what we call our delayed hypersensitivity cells. These are going to initiate macrophages to become extremely hypersensitive, and they're going to become very aggressive phagocytic cells. Again, don't worry about the T helper 17 or the T regulatory. If we have our antigen presentation here, we have tissues, we have cells. Our T helper 1 can go and initiate with the release of interleukin 2. We can initiate some activation of our CD8 cells here. But let's look at our cell here. This is going to be a tissue cell, whereas this is a cell that we're going to find in the blood. This is a tissue cell that carries an MHC1 class 1 receptor. It's placed that antigen in its antigen binding site, and now we have our CD8 cell, which is going to bind to this antigen, and we're going to activate those CD8 cells. Those CD8 cells then are going to differentiate and mature into our cytotoxic T cells. Our cytotoxic T cells are going to recognize the antigen. They're going to bind and they're going to produce these perforins, these granzymes, and they're going to go then and destroy that infected cell, and that cell then is going to die in the process. So you can see here is essentially the whole activation of our immune response. If you can just follow through and see where all of these are going to be going. So we have our T memory, both from our CD4 and our CD8 and our CD4 receptors. We have our B memory cells through the activation of our B cells, and so we have a lot of memory that will help us when we have an immune response on a second or subsequent exposure. I know this is very complicated and it kind of has gone around in a circle, and I've tried to follow the book as easily as I can. You do need to take some time to kind of go through this with your book and go through this with these pictures. This picture right here is on page 442 in your textbook. I know it says the scheme of T cell activation, but this is essentially your immune response, your humoral and your cellular immune response in a nutshell. If we look here, you've got your T cell activation, your CD4, which is going to give you all of your T cells being activated, and one of which is then going to activate your B cells. So here's our humoral response. Here is our cellular response. So this essentially is the immune system in a nutshell. Again, if you're having some questions, please email me so that I can get back with you as quickly as I can. This picture I just thought was a really good picture. This the cell that kind of looks a little bit blue or gray here. This is a cytotoxic T cell that has essentially released those perforins, and you see all these holes now in the cell membrane, and it's being destroyed. So this is kind of looks like a David and Goliath moment, if you will, because if you look, that microbe is much larger than this small cytotoxic T cell, and that cytotoxic T cell has basically destroyed it just by punching holes and tearing apart the membrane. Very cool picture. We do have a set of cells that I haven't really discussed much, except when I introduced that picture of the division of blood cells that come from the hematopoietic stem cells, called the natural killer cells. And these are related to the T cells, but they are not as specific antigenically as the T cells are. These are going to circulate through particularly the spleen, the blood, and the lungs, and their job is cancer surveillance. That's their first and foremost, and they're also going to be very helpful in virally infected cells, in the detection of those virally infected cells. So this really is pretty much all I want you to know about the natural killers, that their job is mostly cancer surveillance, and they are going to help you out throughout the infection process through viruses. So here is the immune response in a nutshell. This is a very good animation. I would encourage you to watch it several times, go over it several times, listen to it several times, turn the words off if you can, just pause it, turn the audio off, so that you can go over it numerous times and see what is happening with your specific acquired immunity.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
So watch this over again a couple of times. Stop it between the cytotoxic exposure and the B cell exposure, so that you can differentiate that cellular immunity, those cytotoxic T cells, and your humoral immunity, which is the production of your B cells and antibodies. So again, I can't encourage you more. Please, please, please send me any emails so that I can answer them before you need to come and take the exam. So now that we've talked about the immune system and how the immune system functions, how that it gets triggered and is initiated, we can put our immune system, we can kind of classify it with four basic descriptors. It can be an active response, it can be a passive response, it can be a natural response, or it can be an artificial response. When we talk about natural versus artificial, or passive versus active, this active response can be natural or active. You're going to activate the B cells and the T cells, and when you have an active response, you are going to produce memory. So you have long-term protection through these active responses, regardless of whether they're natural or artificial. When you have a passive response, these can also be natural or artificial, but you're going to receive antibodies from another source, whether it be a human or another animal. Because the antibodies are preformed, there is no memory. Remember, antibodies come from plasma cells, and the plasma cells are not memory cells. You have no antibody production because, again, the antibodies are preformed. So this is going to be very short-term protection. You have no memory here. Natural immunity, I said, could be active or passive, and natural active immunity is going to be direct exposure due to a natural process of disease. You've gotten sick, you've breathed in, or you've taken in some pathogen through some portal of entry, and you have gotten sick. You're going to develop a very specific targeted response. You're going to produce memory, but you are going to suffer the consequences of illness. Okay, you are going to get sick. When you have a natural passive response, this is going to be antibodies that are passed through the placenta or through the breast milk, IgA and IgG antibodies specifically. Because this is a passive response, you're getting it from one person to another. The antibodies are already produced. You have no memory here. This is passive in the fact that the person gaining protection is not producing the antibodies themselves. But this is called our natural immunity because these antibodies are given through a natural response. Artificial immunity, think of it this way. If it involves a needle, it is completely artificial. So we have our artificial active and our artificial passive immunities. Artificial active immunity is the immunity that comes as a result of some sort of immunization or vaccination. You're going to be artificially exposed to an antigen, and this is going to stimulate antibodies and a memory response. Artificial passive immunity is also going to entail injection. You're going to have the injection of some sort of serum from some other patient, and this is going to be an attempt to neutralize whatever the threat for that particular antigen is. A lot of times this is going to be some sort of viral attack. You may have antibodies from some sort of viral attack. This is going to be therapy. This is going to be immunotherapy. There is going to, again, be no memory. You are not producing antibodies because you are getting the antigens, the antibodies already preformed in this passive response. So if we look here, we've got our natural active immunity. This is a consequence of an immune response to some sort of microbe. We have our passive immunity, our natural passive immunity. This is one person receiving preformed an immune response made by another person. This is going to be acquired through, these two are going to be acquired through a normal life experience like illness and congenital things like across the placenta or breastfeeding. Artificial immunity or artificial active immunity is going to be a consequence of that vaccination. I don't know any kid that is quite that still for a vaccine. But anyway, you have this artificial active immunity, and then you have artificial passive. This is going to be a person receiving preformed antibodies made by another person, so that whatever they are sick with is going to make them better, and they are going to receive protection in a much more rapid response than if they had to produce the immune response by themselves. Antitoxins are essentially antibodies that are going to neutralize toxins. These can be given in the form of artificial passive immunity. Think about rattlesnake venom. These antibodies are preformed and can attack those particular toxins. These antitoxins can also kill bacteria, or excuse me, can neutralize those toxins, but you will use antibiotics to kill the bacteria that produce the toxins. You are going to have these antitoxins eliminate any sort of circulating toxins so they can't bind to tissues and cause further damage. Once you've applied the antitoxin, then you can give the antibiotics, kill the bacteria so they're not producing any more toxin, and you are going to get better. These antitoxins are going to be particularly helpful in things like tetanus and botulism. Let's talk about vaccines. This is going to be part of our artificial active immunity, and we have some principles that we need to kind of follow for the preparation of a vaccine. It needs to be selective against a particular antigen. It has to have, we need to have good effectiveness. Does it kill the microbe that it's targeted for? Ease of administration. Can it be given with a shot, or can it be taken in the mouth? Is it safe? Do we have any things that could complicate the patient's health? And are they cost effective? If you look here on table 15.4, you have a checklist of the requirements for an effective vaccine. Take some time to read over those so that you can be familiar with the responses of a vaccine. Vaccines can be made with a number of different preparations. You can have whole cell or viruses. You can have, when you have these are what we call live attenuated cells or viruses. Live attenuated essentially means that the cell is living, but it has been dampened down to potentially not make you as sick as it would if it were a full-blown cell or virus. Or these can be what we call killed cells or inactivated viruses. These can also be ways that you can prepare vaccines to get an antigenic stimulation. You can have some of these vaccines where you're going to have molecules from the antigen. They are going to be used as what is called a subunit or cultured vaccine. These subunit vaccines are going to take cultured cells or viruses, and they're going to use just a portion of them to make the vaccine. Or we can have what are called synthesized subunit vaccines, which are only going to, again, use a small fraction of the bacterial cell or virus. Inactivated versus attenuated. There's some argument about the safety of some of these. When you use dead organisms, these are going to be the safest because they cannot cause disease. Generally, they are going to be less effective as immunogens because they're dead. They're not producing any kind of antigen, they're not producing any kind of toxin, and they are moving in through the wrong portal of entry. If you have a respiratory cell going into the bloodstream, that's not the correct portal of entry. So you're going to have them be less effective, but they are safe. Live organisms are going to give you a much better response. These are going to mimic the actual infection. Think about the FlowMist vaccine that you use for the flu. You take that in through the nasal passage. This is a live organism, and it binds to those cells and gives you a very strong immune response. The problem with these is that they can mutate back to their virulent form. We have a whole population of people that have a mild case of polio due to the Sabin oral polio vaccine. That vaccine mutated back to the virulent form, and so now you have a population of people that have polio. This picture is a great picture on vaccine design. It basically shows you some of the ways that you can take these organisms. If we have a killed cell, we can heat it. Then we have the antigens from those, and we produce a response to that dead cell and we're going to produce that chemical or that vaccine. You also have these live attenuated cells. The virulence is basically eliminated or reduced. You have these act now as antigens. You're going to administer those as a vaccine, and they're going to stimulate an immune response. But again, they are not going to give you any disease. So these are what we would call our whole cell vaccines. Our subunit vaccines, you're only going to take a portion, maybe the spikes, like we do with the influenza vaccine. You're going to take those spikes. Those spikes then are going to be the thing that the immune system recognize. This is going to be what your vaccine is going to be made of. Or you're going to take some of the pieces of a bacterial cell, maybe the flagellum, or some of the intracellular proteins, the cell membrane, the cell wall. Any of those kinds of things can act as a subunit or a piece of the antigen. Those are going to be the vaccine, and those will be injected into the patient and will stimulate immunity but no disease, because you have no pathogen present. Vaccines, companies that are still putting some time and effort into vaccines, a lot of them are looking towards these subunit vaccines as the way to go, because they do give you a relatively good immune response, because you are getting portions of the live the live bacteria or the live virus, and it's giving you a pretty good immune response, and it's not going to cause disease because you only have a piece of the pathogen and not the entire pathogen. The development of new vaccines is relatively difficult, especially since a good portion of the host immune response is not really clear as far as the infection process is concerned. You have a number of these viral, you have these latent viruses, you have these persistent viruses. The host immune response doesn't always give you a very clear picture, and that artificial immunity has to outperform the host response immunity to have any sort of real effect. So sometimes that natural response is going to be a better response, and therefore the vaccine isn't really as effective as it needs to be. It is difficult to choose a vaccine for antigens that are safe. You have to be sure that if you're going to use a whole vaccine and it's going to be a live attenuated, that you can attenuate it so that it will properly stimulate the immune response without causing infection. Or if you're going to have a killed vaccine, is it going to give you a significant immune response to give you that proper stimulation? Same with the subunit vaccines. Research now is going to use some of those strategies for vaccine production by what we call recombinant DNA technology. Where we're going to take a piece of the chromosome of the pathogen, combine it with something, the DNA of something that is avirulent or not disease causing, and we're going to then use that to produce the protein that the body will recognize as the antigen. The protein is not going to hurt you, but it is going to stimulate an immune response. We're also looking at gene cloning technology. That gene cloning technology is going to allow us to clone the gene that's important in the pathogen without cloning the entire pathogen, so that we have just the gene and just the protein that comes from that gene, again giving us proper stimulation so we don't harm the host. You have some genetically engineered vaccines. These are going to be our recombinant DNA technology vaccines. These are what we call Trojan horse vaccines. Basically, you're going to take the genetic material from the pathogen, and it's going to be inserted into something that is non-pathogenic, like a yeast cell. The recombinant microbe then is going to multiply over and over and over again. It's going to express that foreign gene, and that foreign gene then is going to express the protein, and the vaccine recipient then will be immunized against the microbial agent without having the agent present. So we also have our DNA vaccines, where we're going to take that foreign DNA and insert it into a host. a foreign antigen is going to be produced, and then we're going to be protected by the immune response. If we look here, we're going to take a piece of the pathogen, we're going to insert it into something like a plasmid, and that plasmid then is going to be inserted into that vector, that plasmid vector. This is going to be amplified. What we mean by amplification is we're going to make copy after copy after copy. Once that has been amplified, now we're going to take that DNA vaccine and inject it into the host. That plasmid now is going to insert itself into the human cell. It's going to be into the person's cell, and that DNA in that plasmid is going to be transcribed and translated, and it's going to be injected into translated into the host chromosome, and you're going to end up with all of these particular proteins of the pathogen on the surface of the cell. And this is where you're going to stimulate an immune response. That immune response now is going to target the proteins that are produced by the pathogen, and we will then produce an immune response. Here is a good animation on how they construct vaccines and the ways that vaccines are going to be, people are starting to look at vaccines in the future.
There are a variety of ways to produce vaccines. Traditionally, vaccines have consisted of killed or inactivated pathogens. A second traditional approach is to use an attenuated strain of the pathogen. During attenuation, the pathogen loses its virulence, but retains many of its antigens and is therefore able to elicit an immune response. Subunit or acellular vaccines consist of selected components from the pathogen, such as a surface structure, an internal macromolecule, or the flagellum. Recombinant vaccines are produced by cloning the gene for a structure, such as a surface protein from a pathogen, into a host microorganism. For example, the gene for a surface protein of the hepatitis B virus has been cloned into a yeast cell. The yeast produces the virus surface protein, which can be purified from the growth medium of the yeast and used as a vaccine.
So you can see that we can use either the whole cell in its dead state, in its living state, or we can do those subunit vaccines, which are going to give you a strong immune response without the pathogen. So most of our vaccines from here on out are probably going to be those subunit recombinant vaccines that are going to give a high level of immune response with a low level of side effect. Administration of vaccines is always going to have its particular side effects. Most vaccines are going to be injectable. They're going to be either subcutaneous or intramuscular. Some of them do have interdermal administration. Only a few oral vaccines are available. As a matter of fact, I'm not really sure of any right off the top of my head that are oral anymore. The polio vaccine in 1998 was discontinued, and now they're back to the injectable form. Some of these require what is called an adjuvant. An adjuvant is something that needs to be added to the vaccine and will help with the administration of the vaccine. You do have some side effects, some common side effects that you have because you are producing an immune response. You are going to have a potential local reactions at the injection site. A normal inflammatory response is always going to be expected at the injection site because you've broken the skin, and the skin now is going to produce that inflammatory response. But if that inflammatory response becomes overwhelming, you begin to produce a great deal of swelling, the rash gets larger, you do have to take that into consideration and get some treatment. Some of these will produce fever. Some people will in a few days begin to mount a small low-grade fever, And then allergies. Some people are allergic to the preparations of of vaccines, and they have to have some other way of administration or be exempt from that vaccination. Some people do experience some reactions to the production of the vaccine rather than the antigen itself. All of these are made up into a solution, and they are allergic to the solution, or they have a reaction to the solution rather than to the antigen. But for the most part, most vaccines are perfectly harmless. They give us great protection. And when we choose not to vaccinate our children, we kind of put them at risk, as well as the other people in the community at risk, because whatever we are vaccinating against is out there in the community, and you are going to have some sort of response when they become infected. So vaccination, in my opinion, is always a good thing. To vaccinate, do we not? Vaccination does give us protection, and it does keep us from becoming sick from some of these different microbes that we have all the time. But not only does it keep us sick, it also gives us a good sense of public health. When you've got when you've got a large number of people who are unvaccinated, they can pose a significant public health risk to people who are immunocompromised, who are very young, very old. So you have to think about those kinds of people, those kinds of community issues when you choose not to vaccinate. There is something called herd community, and basically this is collective immunity through mass immunization. This is going to confer some protection on non-immune members, basically because what has happened is you have a number of people that have become immunized, that there isn't a niche for this organism to live anymore, because it doesn't have a place where it can infect and grow and replicate. And so because there isn't a niche in that community, those that are non-immunized gain the protection from those who have been immunized. This is very influential in preventing epidemics of these of particular pathogens. For instance, diphtheria. We don't see diphtheria in the United States anymore because there is no niche for it. We get the DPT vaccine when we're small, and because there isn't a niche, we kind of have this herd community of protection against diphtheria. This is just a picture of some recommended vaccines that we should get from the time you're born throughout your life. Most of us stop getting vaccinated when we're probably middle school. I know if you're staying in dormitories, in college, in the military, things like that, there are some new vaccines that are required, particularly the meningococcal vaccine. This is extremely important because these are the ones that are highly contagious. In my opinion, everyone should be vaccinated, and we probably should keep vaccinating through our lifetime so that we are not contagious along the way. I do have to say there are some vaccines that I probably would hesitate, like the Gardasil. I'm not really sold completely on that one, but for the most part, I think the rest of them are extremely helpful and keep us in good health individually and publicly as a community.